Okay.
I'm here today with David Crowe. He's the host of The Infectious Myth. He's over the years and recently he's interviewed such luminaries as Dr. Andrew Kaufman, Dr. Thomas Cohen. He even went to California once to interview Dr. Peter Duesberg. He's written a book on SARS, and I believe you're part of a group in Canada reappraising AIDS. Can you talk to us?
Well, Rethinking AIDS is actually a California-based organization. So I'm the president of Rethinking AIDS, and our board contains like an Italian molecular biologist, a German doctor, an Austrian gynecologist, a British filmmaker, an American chemist. So it's a broad range of people around the world.
And you actually went from Canada to Northern California to interview Dr. Peter Duesberg in his lab. What was that like?
Well, Peter is an amazing person. He's eccentric. He's charming. He's smart. He's like very focused on his work. And his work right now is cancer, because he understands that he's been so blacklisted in studying HIV and AIDS that there's no way he could ever get anything published anymore. But I mean, with Peter, it's like he's an interesting human being as well as everything else. Like I walked from his house to UC Berkeley with his dog, and we're having conversations with his dog and all those kinds of things, as well as having conversations about molecular biology and politics and everything like that. So yeah, he's just a well-rounded, interesting person, and with a lot of courage, because he's living in a modest house in Oakland. He could be living in a mansion, as a biotech millionaire if he'd played his cards right. But playing his cards right would have meant giving up on his moral values, giving up on what he thought the truth was. And so he's stuck to his guns on HIV and AIDS, although he's not really involved on a daily basis because it's kind of pointless.
Yeah.
He still stands by his contention that was published in his 1996 book, Inventing the AIDS Virus, that HIV is not the cause of AIDS.
Yeah.
Now he believes that HIV exists, but he thinks that it's in such small quantities that it's not pathogenic.
Yeah, in fact, you mentioned that great book, Inventing the AIDS Virus, and it's interesting because Kary Mullis, who invented the PCR test, which is now being used to diagnose and corona supposedly and grant or deny privileges in society worldwide. Kary Mullis, the inventor of PCR, actually wrote the foreword to Inventing the AIDS Virus. Is that correct?
Yes, and in that foreword, it's interesting. He must have been working on some papers on PCR, and he was probably told, we're going to use PCR on HIV. And so he said, okay, if we're going to write a paper on it, he wrote down HIV is the probable cause of AIDS. And then he thought, okay, well, I need a reference for this. But that's easy because there's like 5 million AIDS researchers. So I'll just go talk to some of the people I know, and they all kind of fluffed him off or talk to somebody else. And so he kind of went up the food chain, and he eventually talked to Luc Montagnier. And Luc Montagnier couldn't help him. I mean, Luc Montagnier got the Nobel Prize for discovering HIV, and yet he can't come up with a paper that conclusively proves that HIV is the cause of AIDS. And I don't know. Later in his life, Kary Mullis apparently kind of went quiet because I think he was warned that he couldn't work in the biotech industry if he kept being so outspoken. Yeah. So we don't really know what his views were at the end of his life.
Yeah. Seems like if he still were alive, he'd probably be critical of the way the PCR is being used for coronavirus. So we want to get into that in a little bit, but sticking to AIDS, I was listening to your Infectious Myth podcast just yesterday with Dr. Rivington Nevin talking about how dangerous these antiviral drugs can be. And it seems like that's true also for ACT, but the side effects are overlooked because it's assumed that AIDS patients have to die. And that's kind of where Dr. Duesberg comes from as well.
Yeah. And what's really interesting is that remdesivir is from essentially the same family of AIDS drugs as AZT. And the mode of action of AZT is it interferes with DNA synthesis. So DNA, for people who've taken high school biology, is made up of 4 beads on the chain, ACGT, adenosine, guanine, thymidine, ACGT, I missed one, cytosine, I think
Okay.
I missed. So there's 4 beads on a chain, and the T in AZT stands for thymidine. So it is a fake thymidine bead, but it is a broken bead. And so you get one molecule of AZT into a growing DNA chain, and the DNA chain will stop growing.
And it's true that in order to have followed scientific protocols, correct me if I'm wrong, they would have needed to have taken a healthy group of HIV positive patients and not treated them and then compare them to the treated group. But they didn't do that, and there were risk groups. And
well, they claimed to have done it in the initial AZT trial, but there were 2 people who played an important role in showing that was not true. One of them was John Lauritsen, who was a gay man who was opposed to the use of AZT, and also was an anti-poppers activist, poppers being a recreational drug used by gay men. And the other was a woman named Lynn Fall. She changed her name when she got married. I can never remember which is the current name. And she was like data processing for the trial. And they both found evidence that the trial was completely corrupted. Some ways were corrupted inside the trial, some outside. For example, the patients were so desperate to get drugs that they mixed their drugs together. So you might have had 3 patients, and maybe 2 of them had AZT and 1 had the placebo, and they mixed them all together and just randomly took pills. And so now they're all on a lower dose of AZT. So, I mean, that completely destroys the trial. They found evidence that people who got sick were moved from the active arm, the AZT arm, to the placebo arm. There are all kinds of things that went on. Mm-hmm. And, you know, AZT is also, like a placebo for a drug as powerful as AZT is very difficult, because you know when you're taking AZT, you can feel the effects on your body pretty quickly.
Yeah.
So you're going, okay, I'm feeling really sick, so I must be taking AZT. But there's you want the trial to succeed. So there's kind of a human, I'm going to minimize my side effects, you know, when I'm asked about them, because I want people to get access to this drug. You know, it's counterproductive and it's unscientific. But how do you come up with a placebo that has to be a dangerous drug, or you can easily tell that you're not on the placebo?
Yeah. And with the AIDS defining illnesses like Kaposi sarcoma, this kind of skin cancer, it's known that this is a side effect for the type of nitrate inhalants that the gay community was doing. So there's also,
yeah, I mean, two important people there, John Lauritsen, and he had one other guy who worked very close. Hank Wilson worked closely with him. Unfortunately, Hank Wilson is now deceased. But they knew that poppers were dangerous, and they tried to educate the gay community that these are dangerous drugs. Another important man is a man named Harry Habermas, who is a completely mainstream AIDS scientist. He told me, I interviewed him recently, and he told me that he came up with the first definition of AIDS. So like, this is a guy inside the establishment, but he believed that poppers were the cause of Kaposi sarcoma. Now, he also tried to say that HIV was involved and stuff, but that part isn't terribly convincing. But the fact is that even today, he still believes that poppers cause Kaposi sarcoma. And poppers are immunosuppressive and they're carcinogenic, so they match the type of diseases that are classified as AIDS. Now, of course, poppers are not used by IV drug users. They're not used by Africans. They're not used by transfusion recipients. So there's this crazy belief that every disease has a single cause. Now, if a disease was very precisely defined, like let's say that you have a disease where everybody has fever, rash, vomiting, their eyes turn yellow, their hair falls out. Every single person has those. You could say, okay, well, there's probably a single cause. But what happens is you define a disease with those symptoms. And then it's like, okay, if somebody has their hair falling out and yellow eyes, they match the disease. And if somebody has a fever and an upset stomach, they match the disease. And before you know it, it's like pretty much everybody in the world who's ever sort of been mildly ill matches the definition. And AIDS was like that with 30 different conditions that they just kept throwing into the mix. They threw a couple in, like cervical cancer, that are only found in women, just to try to make it an equal opportunity disease. Didn't work, but they tried.
Right.
But there was no connection between the diseases in the definition of AIDS, except HIV. And so if you question the role of HIV, not just does the HIV theory fall apart, but the idea of AIDS as a definable syndrome falls apart.
Who questioned this?
Well, I'm just saying that if you question the role of HIV and AIDS, you're also questioning the existence of AIDS. That doesn't mean that people don't have the individual diseases, but the only thing that glues AIDS together, because all the diseases are previously known and occur in people who are not HIV positive, the only thing gluing them together is HIV. So you take away HIV and there's no AIDS.
Yeah. And this concept of actually looking for a virus at virus isolation, that's something that you talk a lot about in your podcast. And during this time of lockdown, when we've all been out of work, I've been listening to your podcast, and it's been really educational. And as I've been questioning COVID-19, it's easy to then question, well, what about this virus, and what about that virus?
Yeah, I can only examine so many viruses, but every virus I have examined, I've come up with similar flaws in the reasoning. And I've also found, interestingly enough, alternative environmental explanations for the illness.
You wrote a book on SARS, is that correct? And I think you mentioned
a book chapter is.
Oh, a book chapter.
I think we're going to publish it. It's not published yet, but I think we're going to publish it as a standalone book, and then we'll publish a bunch of books, and then later we'll kind of put them all together into one coherent book. I'm thinking 6 or 8 different diseases to try to show that criticisms of infectious diseases have a lot in common. I think it's important to understand. And with the flaws are similar.
With SARS, I heard you mention that was like the trial run for treating flu with antiviral drugs. SARS is supposed to be a coronavirus. So it's the closest thing that we know of to COVID-19. SARS caused the shutdown of a portion of the world, parts of China, Hong Kong, Taiwan, Singapore, somewhat in Vietnam, and Toronto in Canada. They were locked down pretty hard. So that was kind of a trial run. They used intubation a lot with SARS, just like they did at the start of the COVID-19. They kind of backed off on that. They used antiviral drugs a lot. Okay.
With SARS, there was only one antiviral drug that they used a lot, which was named ribavirin, which is similar to AZT. And all of the interventions that they used, they found afterwards were ineffective and dangerous.
How were they playing with the numbers? You said that in big cities, there's so much false positives, even admitted false positives, that they can cause an epidemic just by focusing on those. Well,
SARS was a little saner. Just like COVID-19, a probable case was like a fever, 38 degrees, 37 is normal body temperature. And, pardon me, fever and epidemiological contact. And that was really important because that limited the scope of the definition. With COVID-19, they basically said, if you've got a positive test, you've got COVID-19, screw the epidemiological connections, screw the symptoms. Mm-hmm. So now you have this massively greater number of people who are testing positive. With SARS, it was kind of like, if you don't have a symptom and you don't have epidemiological contact, we're not going to test you. Now they test everybody.
But the existence of COVID-19 is based on assumptions about the existence of SARS.
Yes, because it's a coronavirus. So they looked at the RNA and they said, oh, this looks a little bit like SARS RNA. So we're going to call it a coronavirus. And that's how this whole thing got started. But they never proved that the SARS RNA actually came from a virus. So we still haven't gone to fundamental step one, which is the 30,000 base RNA sequence from a virus? And the only way to determine that is to purify the virus. And they can't do that. I mean, unless you have pure virus, you don't know that what's inside the particles you're looking at is the RNA that you're working with. So in papers where they have pictures of particles and they have a description of the RNA, you really have two completely different things, and they shouldn't be in the same paper because there's no proof that those particles have RNA in them to start with, let alone the RNA you're looking at. There's no connection between those particles and the RNA. And with the RNA, there's no proof that that RNA came from a particle of any kind, let alone a virus particle. And so by putting them into the same paper, it's insinuating that these things are related, but they're probably not.
Why don't you give us a rundown of your critique on the COVID-19, how they're fudging the numbers, and then maybe we can get into the PCR test and why that's such a fiasco.
Yeah. So with COVID-19, they're basically saying everybody who's positive is infected. And that includes a majority of asymptomatic people. They are, in some cases, including people in the counts who are just cases they think are positive. Like in Belgium, for example, they define every death in a nursing home as COVID, even though something like 5% of people in nursing homes have been tested. In a nursing home in Brooklyn, New York, where 55 people died, nobody in the nursing home had been tested. In Ontario, Canada, if one person in a nursing home tests positive, the nursing home is considered an epidemic outbreak, and everybody in the nursing home is considered a probable case. So the numbers are quite meaningless. I think a CDC or some other organization in the US admitted that gunshot victims had been included in the COVID statistics. And you know, that's just kind of funny because it's only 5 people, but it's like how many other people? It's important to realize, I guess the Italians have the best statistics on this. They studied 26,000 deaths. They come up with an average age of 80, which by itself, you know, is a lot of healthy 80-year-olds. 60% of these people who died had 3 or more pre-existing health conditions, which included kidney failure, liver failure, heart failure, you know, heart disease, diabetes, severe obesity, things like that. So the people who were dying were extremely sick to start with, but that doesn't mean they died of COVID. There was a scientific advisor to the Italian health ministry was interviewed by the Daily Telegraph newspaper in the United Kingdom, and he admitted that only 1 out of 7 of those people that they had recorded as COVID deaths had actually died of COVID. Others had died of heart failure. They died of kidney failure. They died of, you know, their heart was weak and they were so obese that their heart just gave out. Also, another, there's a lot of iatrogenic or doctor-caused causes of death, such as intubation in New York.
What's that mean? Tell us what that means.
Okay, so intubation is a really horrible process. I mean, most people think, okay, you stick a tube down your throat and it helps you breathe. Well, I want to stick a tube down your throat. Are you going to let me? Open wide, and I'm going to stick a tube down your throat. You're not going to let me because your body has reflexes. Apart from your conscious resistance, you have subconscious resistance to having anything put down your throat. So they have to paralyze you. They don't just sedate you.
Why do they stick this down your throat? Is this for the therapy?
To get the tubes into the lungs.
Is this the ventilator thing?
Yes, the ventilator, intubation is the same thing.
Okay. Yeah, I heard that instead of using oxygen which might have been more sensible, they started using ventilators like overmuch. Is that where you're talking?
Yeah, yes. And here's the really shocking thing. The reason they put people on ventilators is because ventilators are a closed system. The air goes in and out of your lungs without going into the room, right? You're not breathing on the ventilator. The machine is breathing for you, and it has a tube going in and a tube going out. So it is a completely closed system. So the reason they were ventilating was because they were scared of infection. And I think this is literally criminal behavior, because you are not doing something for the benefit of the patient. You are doing something for your own perceived benefit. During SARS,
New York overbought like 300% worth of ventilators.
There are warehouses full of ventilators that will never be used because they realized they were literally killing almost everybody.
So
in New York,
the process of putting it in the lungs can be very damaging to the person.
That's right. There are conditions called ventilator-associated pneumonia. So the ventilator creates pneumonia, which of course you're giving people ventilation because they've got pneumonia. How do you know which one is the cause, right? The second thing is ventilator-associated lung injury, VALI, which is a syndrome of different illnesses that are all associated with ventilation. But the bottom line is that in New York, of the people over 65 who were put on ventilators, 97% died. A Chinese study found the same 97% death rate. On people under 65, the death rate was 76%. So ventilators are actually death machines. And they knew this with SARS. They knew this in 2000, maybe not 2003, but in 2004. There was a study that looked at 14 hospitals in Hong Kong. 13 of them routinely ventilated everybody. And they did this for fear of infection. They did not do this for the benefit of the patient. Those hospitals had about a 4.5 times higher death rate than the one hospital that only ventilated when necessary. And that hospital found that ventilation was rarely necessary. It was also interesting that not a single healthcare worker in the one hospital that didn't routinely ventilate came down with SARS. So the fear of infection was misplaced. But they killed a lot of people during SARS with ventilation. And did people learn from this? No. They went and did exactly the same thing with coronavirus, and they had to learn for themselves. When the doctors in places like New York started to see every patient dying, the doctors started to say, well, this isn't what we expected. But you're not just taking an average person, an average healthy person who, or reasonably healthy, maybe somebody's having an operation and they need to be ventilated for a few hours. What we are doing we are taking the frailest people in the world, an 80-year-old person with 3 pre-existing health conditions. They got a weak heart, they got a weak kidney, and you put them on a ventilator, which stresses all your organs. Mm-hmm. Like the death rate is going to be higher. It's going to be close to 100%.
Wow. So this is iatrogenic illness, which means doctor caused, and then it goes to inflate the numbers for the.
Yes. So everybody that the doctor kills gets counted as a COVID case.
Wow.
And so the numbers of COVID deaths are meaningless. Mm-hmm. The number of COVID infections is meaningless because we don't know that the test actually tests for infection. So, I mean, other tricks they're playing is in some states in the United States, they're putting people on the COVID list if they test positive for RNA or antibodies. But antibodies are supposed to say that you've cured yourself of the virus. So you're not actually a COVID case. But they just throw them in anyway.
You wrote an article recently about the antibodies, and that's something that I wanted to ask you about. But before we do that, I just want to talk about the PCR, because people in Ecuador now, in order to even come to the country, you have to have a PCR test, and it's got this godlike status. But as I've been listening to you and others talk about how they do all these amplifications, and there's nothing to compare it to. It hasn't followed the scientific protocols, and they don't even know what they're testing for because they never isolated the virus. Can you talk a little bit about PCR?
I don't really know where to start. There's so many things wrong with it, but I'll give a little bit of information on the technology and talk about, why we know there's problems with it. So PCR was invented as a technology that could take a strand of DNA, could find the part of that DNA that you wanted to replicate, and could, in a day, create 1 billion copies. So it was like a photocopy machine for DNA, but it was better than a photocopy machine because it's kind of like you tell the photocopy machine, I want this one paragraph duplicated. And at the end of the day, you have 1 billion copies just of that one paragraph, not the whole book, right? So it was a very good manufacturing technique. And so people said, well, if we have an unknown amount of DNA in somebody, and we run this process, at each step, the amount of DNA doubles. So if we calculate how much DNA we have after, say, 20 cycles, that is 2 to the 20th times what we started with. So it's simple mathematics. A problem with RNA is that the test doesn't work on RNA, so you have to convert the RNA to DNA. So there are 3 phases, each of which has problems. The first is you have to extract RNA from the sample. You have to get rid of the DNA in the sample, and you have to get rid of anything in the sample that could actually interfere with the test.
And this
There's
was
lots of stuff.
This is where they were adding antibiotics to the samples.
No, that's isolation. They do this. Yeah. So this is like a nasal swab. You try to extract the RNA. Well, first of all, you can't extract all the RNA. You can't get rid of all the DNA, and you can't get rid of all the interfering substances. So the amount of RNA you end up with at the end of this process is less than what you started with. So you already have a quantitative problem, right? You don't know how much RNA you actually have in the original sample because you didn't get it all. The second phase is that you convert the RNA to DNA with an enzyme called reverse transcriptase. But according to a world expert on a technology who I interviewed,
I heard that.
This is less than 50% efficient. So now you take the small amount of RNA that you got from the sample, which is not 100%, and you run it through a process that is less than 50% efficient. And he said the efficiency varies by a factor of 10. So now you have no clue how much DNA you have relative to the RNA you started with. There's no connection between these 2 numbers.
And it's contributing to the panic. And like you eloquently noted, people get confused by the panic. They hear there's all these cases here, all these cases there. They don't even know what that means. And
well, that's true. And they believe the results of this test. The third phase is you cycle through PCR, and at each step, you approximately double the amount of DNA you have. The DNA that is the mirror image of the RNA.
The amplifications that also amplify the noise.
Right. So you can do the math on this, right? If you do 10 cycles, you have 1,000 times more than you started with. You do 20 cycles, you have 1,000,000 times more, et cetera, et cetera. Except that the process is not 100% efficient. And except that you don't know how much RNA you started with. The amount of DNA you have bears no resemblance. And so how do they distinguish between positive and negative? Well, they use a quantitative cutoff on a test which is not quantitative. So they use between 30 and 45 cycles. And if you can detect the DNA you're looking for before that number of cycles, you're positive. And if you can't, you're negative. So of the 33 FDA-approved tests that I looked at, which, as I said, varied from 30 cycles to 45, that is a difference in sensitivity of about 30,000 because you double it each cycle. The tests are looking for different genes. So a gene is just a portion of the genome. The portion of the genome they're looking for is usually less than 1% of the entire genome. The way you decide if somebody's positive is different. For example, some tests look for 1 gene, some look for 2, and if they look for 2, some demand that you have both. Some say either one is okay, and some look for 3. Some demand that you have all 3 genes. Some say 2 out of 3 is good enough.
Okay, sorry.
Which brings up a question of how could you have one gene from a virus and not the other?
Yeah.
It doesn't make any sense.
Have they ever tested samples from last year before COVID-19 was around to see if they get positive results?
I don't think they've done it with the RNA test, but they've done it with the antibody test. And since you should only test positive on an antibody test if you've been infected, then blood from 2018 cannot be positive on antibody tests. But in a Dutch study, they found that 14% of samples were positive for antibodies from 2018.
That's a lot.
So that's impossible. So either the virus was around in Holland in 2018 without causing any noticeable problems, in which case we just panicked for nothing, or the antibody tests are highly inaccurate, which I think is the more likely case.
Why don't you talk to us about these antibody tests? Because I know you've thought a lot about this.
Yeah, so the antibody tests are supposed to indicate not that you're infected now, but that you were infected in the past. So the antibody test, if you test in, say, the United States for antibodies, the number of people who test positive is the sum total of everybody who's been infected between, say, January and whenever you're doing the tests. Whereas an RNA test, if I test somebody today might be the first day that they're positive, or today might be the last day that they're positive, right? It doesn't tell me anything about tomorrow. So you would expect antibody tests to have a much higher percentage positive than PCR tests, but it's the opposite. When they test people in the United States, they get 10 to 25% positive routinely for the RNA, but they only get 1 or 2 or 3 or 4 or 5% positive for antibodies in most samples. So the numbers don't seem to work out. It seems like there's a lot fewer people who have antibodies than should if these are truly antibodies related to the RNA, which maybe they're not.
Yeah. In Ecuador, I know there's a lot of confusion, and people say, well, what about the dead people? But the chaos that the hospitals went in contributed to the dead people, like the private dialysis clinics closing. And
I just wrote an article, it's not published yet, on what I think is the most serious part of the lockdown, which has got nothing to do with you. You know, all the inconveniences to you are not significant compared to what I'm going to talk about. And that is banning visitors from old folks' homes. Why is this important? Well, we've discovered in Canada, when soldiers went into old folks' homes, or nursing homes, we'd call them, to try to help because of staffing problems, things like that, they found a shocking list of horrible things, such as highly unsanitary practices being used. Old people being left in bed, in one case, for up to 3 weeks without being moved, which is guaranteed you're going to get bed sores if you do that. People not being fed properly. People being fed through a tube, but the food was in the bottle for so long, it had gone rotten. Stacks of trays of food stacked on the floor long enough that they'd gone moldy. Staff abusing patients. The list, this report by the Canadian Forces is truly shocking. And the absence of visitors is part of the problem, because visitors perform a number of functions at a nursing home. First of all, they observe the status of their loved one. So if their loved one has a wet diaper when they come to see them, they're going to complain. If their loved one smells like they haven't been washed for 2 weeks, they're going to complain. So first of all, visitors are going to significantly increase the quality of the place by complaining about things. Secondly, visitors take over tasks from the staff. The visitor will say, oh, I'll feed my grandfather, right? Or I'll get him dressed, right? So that gives the staff more time. So you have staff, many are in quarantine, some have quit, the rest of the staff are overworked. There's no visitors there to take a load off. And the third thing that visitors perform is a social function. They show pictures of the great-grandchildren. They bring a magazine. They talk. But not only have the visitors' social interactions gone, but there are no social events in these nursing homes. There's no group coming in and singing to them. There's nobody giving dancing lessons. You know, they can't sit around and watch TV because they're locked in their room. There's literally no social interactions. I read an article from Germany that I think encapsulates in this one person the whole problem. So this 95-year-old woman was moved to a nursing home in February because she just couldn't look after herself anymore. And of course, the lockdown then happened. And the granddaughter said, well, during the first part, when she had regular visitors, she was doing really well. She was reasonably happy and everything was good. As soon as they cut off visitors, they couldn't phone her because she was deaf. And so they had to send her letters, and she started going downhill mentally and just died.
Wow, that is shocking. And I remember you recently talking with a nurse from England, and
Yeah.
you were talking a bit with him about how the isolation has actually changed the bereavement trajectories of families.
Well, I mean, it's cruel for the family because people die and they might not be allowed to have a funeral. Like this woman said, this granddaughter said that once they phoned and said, it looks like your grandmother's dying, but they still made them wait 2 days before they were allowed to go in and have a visit. And they were allowed one person, 1 hour per day with this dying woman, which is really cruel for the family, but of course it's crueler for the person who's dying alone.
Yeah, that is sad. Just switching gears here. In 2009, we all remember the swine flu scare, but then nothing happened.
Right.
But it's like they hyped up the COVID-19 so much, and they're like waiting for something to happen, and then there were the empty hospitals, and so like now they can't go back and say, "Oh, we made a mistake." I
mean, it's kind of like a kid who plays with matches, right?
Right.
Like he walks into the forest and he throws a match on the ground, and it's like it's cool, and a few pieces of grass burn, and you know, no big deal, nobody got hurt, right? And then he does it again, and then one day he throws the match down, and the entire forest goes up, right? I mean, that's what these public health officials have been doing. They've been throwing matches, SARS, swine flu, and they create a little fire and it burns for a while, but it doesn't do what they want. They have now created the fire to end all fires. And I think what people are finally realizing is that these people are incredibly dangerous and they don't have a clue what they're doing.
Well, you said it finally did what they want. And so that implies that there's another agenda at place besides public health. Can we?
No, I think they have this theory. They have this theory. I hope it's shattered now because it's bullshit, if I could use that word. They have this theory that if you have a new outbreak and you build a fence around the outbreak and you go in there with, you know, Ghostbusters and bazookas and all the weapons you have in your arsenal, and you kill everything inside the fence, that you can wipe out this new infection, right? With all the antiviral weapons you have. This is their theory. Well, how well did the theory work? The virus appeared to infect people magically around the world, but it wasn't actually infecting people magically around the world. What they were getting is false positives. You probably could have gone to a base in Antarctica and tested people in January and found a few positives, because a few people test positive on this completely meaningless test.
And it's meaningless because they never isolated the virus. And this gets into something like Koch's postulates. And then
yes,
it's the sound
but it's worse than that. Let's say that this was a test for a virus.
Okay.
The test is still not perfect. There are still going to be false positives. If you test a country like Italy, and let's say that in February, Italy had no infections, would it have false positives? Well, of course, false positives have nothing to do with an infection. So if you run around Italy testing people, you will find false positives. Now, how do you distinguish that from an actual infection? Well, without purification of the virus, you can't. So we have no way of knowing whether Italy actually had an outbreak of COVID. It seems unlikely, given that the first people who tested positive had no plausible connection with anybody from China. They had not traveled. It seemed impossible that they would be positive, but they were positive. And nobody thought, well, maybe the test has false positives. I mean, it's written in scientific documents that every test has false negatives and false positives. There is no test that does not.
So
and yet they ignore this.
You could take a population of 15 million and create the illusion of a pandemic just by focusing on the false positives. And people get confused by the panic because they hear all these cases and what does that even mean, you know?
Well, let's say your rate of false positives is 1%. So you got 15 million, that's 150,000 positive tests.
Yeah.
So yeah, you do enough tests, you're going to generate an epidemic. I mean, do you want an epidemic of 1,000 or do you want an epidemic of 10,000? All you need to do is do more tests.
Now, getting back to virus isolation, they took, am I correct? They took lung fluid from some patients in Wuhan, but then they were looking for the virus, but they added antibiotics to the sample.
Yeah, well, okay, so my favorite joke here is that you can tell when a politician is lying when their lips are moving, and you can tell when a virologist is lying when they use the word isolation. You would think that isolation and purification mean the same thing, but I'll explain isolation, and then you'll see it's completely different from purification. So you take something like a nasal swab or throat swab or a lung sample or whatever you have, and you want to add it to a cell culture. You have to add antibiotics because you know that there will be bacteria in the sample. So if you add the sample without antibiotics, the bacteria will kill the cell culture. So you have to add antibiotics. You add a bunch of other chemicals to keep the cell culture happy, because the cell culture is cells that will continue to dividing forever. And then isolation is defined as cells in the cell culture dying. You haven't seen viruses. You haven't detected anything about the viruses except that some cells in the culture are dying. And that's it for isolation. As soon as cells die, they claim that they have achieved isolation. Now, purification is based on the observation that viruses are supposed to be protein shells wrapped around DNA, or in this case, RNA, genetic material. And there's also some additional proteins inside the shell. If the virus exists, then you should be able to see these particles. And you should be able to purify them. And the only way to know what's inside those particles, because they're so small, you can't dissect one. We're not talking about a frog. We're talking about something that's like 100 nanometers in size. There's nothing that can dissect it. The only way to know what's inside is to gather a whole bunch together and analyze them. And it has to be pure, because if you do what they normally do, like to take the fluid off a cell culture, that will include bits and pieces of dead cells, bits and pieces of living cells. It could include pieces of dead bacteria, different things like that. And you could be analyzing those non-viral materials when you think you're analyzing a virus. So you have to purify to get to step one, which is to identify the RNA. And you can't produce a test until you know you have viral RNA. But they just skipped by the step of purification. And consequently, we have a test that we don't know is for viral RNA.
Yeah, and when you talked with Dr. Kaufman, you talked about exosomes and how under the microscope it looks similar to a virus.
Yes, so exosomes are essentially identical to viruses under the microscope. It's only their behavior that's different. Exosomes are produced by cells and they're not infectious. They perform some kind of function inside the body, messaging function, something like that. There's lots of theories about what they do. You would have to, if you purified exosomes, and then exposed an animal to them, nothing would happen because they're just exosomes. If you purified a virus and exposed an animal to it, you could cause an infection. That's the only way to tell the difference. And so again, you need purification to know what you're dealing with.
And they've watered down Koch's postulates over the years to Rivers and then Hill, and they keep changing the criteria to lower the bar.
Yeah. I mean, it's kind of like somebody who can't pass their driver's test, who has the ability to rewrite the test. And it's like, well, I have difficulty with parallel parking, so we're just going to take that out of the test and we'll try it again. And you still fail. So then you take out something else. And by the time you're finished, you basically open the door, sit in the car, turn on the ignition, turn it off, get out, and you pass the test. And that's what virologists want for Koch's postulates. Koch's postulates are not scientific. They are logical. And logic of the Greeks is just as valid then as it is today. And if I can restate Koch's postulates, if a virus is the cause of a disease, the virus must exist. If the virus exists, particles of protein wrapped around RNA must exist.
Have they ever been able to find a virus?
I'm unaware of any clear evidence of purification of any virus.
What's that meningococcal? What do they call it? There's different viruses that you hear about, like the tropics.
They talk about viruses all the time, but when you actually look at them, and I mean, I can't look at all the thousands of virus theories that are out there, but the similarities between their logic is stunning. I mean, they don't always use cell death to determine isolation. In HIV, they look for the detection of certain
antibodies,
proteins, antigens, P24, for example. They look for the activity of the reverse transcriptase enzyme. They look for phenomena in the cell culture that are unusual, but that never occur that have never been seen in live systems. There's this giant cell called a syncytium, which is found in some HIV cultures. And if they find that, they say that they found HIV.
Yeah, the whole thing about virus isolation for HIV can be shown to be a joke with films like The Emperor's New Virus. Is fascinating to me. You've probably seen that. I just want to get a couple more questions before I let you go. I want to ask you, how did you first get into doing the podcast? Do you have a medical background, or how did you first get into it? How long?
I have a degree in biology and math, and I realized years later that I was very interested in the sort of the edges of science, the places where things start to break down. I was what's known as a taxonomist, somebody who tries to look at the evolutionary relationships between organisms. And I was working on a hybridization, a natural hybridization of two plants. And I realized that the simple definition of the term species breaks down if you think about hybrids. You know, there's species is this idea that you've got interbreeding between members of a species and no interbreeding outside. There are so many exceptions to this. I could go into it, but the point is that the fundamental unit of taxonomy is a shaky concept. And I found that very interesting because there's so many. Yeah, like if you include hybrids, if you include, maybe methods of gene transfer that might include bacteria, if you include things like lichens, which are symbiotic fungal algae living together as one species, like the term species, it's not possible to define it.
Wow.
And yet it is the fundamental unit of an entire field of science. So I was kind of primed to consider some rather strange theories, because if the evidence takes you there, you have to give up on your, at first as a kid, when I became interested in biology, like species were very simple, and I resisted being told that it's way more complicated and it might not even be definable, right? Like you want a simple answer.
Sure.
And so HIV was a simple answer. And I accidentally heard some people talking about HIV not being the cause of AIDS, and I became very interested. The podcast started, it was initially called How Positive Are You? And I started it with Christine Majori, who was an HIV positive woman who never took drugs, who was healthy for, I think, 16 years before she had a couple of tragedies in her life, and she eventually died. And then in 2014, I started the podcast, How Positive Are You? On Gary Null's PRN.FM. But by then, I had done a tremendous amount of research. I'm also involved in computers, and I developed a database, which is now well over 12,000 documents. And so every scientific paper I read went into my database. And not only that, but I extracted from the paper that I read the most important sentences, 2 or 3 sentences that encapsulates what's important about this paper. And from that, I was able to create lists. So for example, if you take the drug AZT and you want to know about the problems it causes for blood, I have a list for that. It's a list of scientific papers with an extract from each paper that tells you what's important. It tells you what blood damage was found in this research. And so I was able to use my computer background and my biology background to create a database that made it possible for me to, it just made the research that much more useful. If I find a paper on SARS when I'm not researching SARS, it goes into the database, and then later I will use it, right? So I don't lose anything that way. And I might find a paper that I'm not sure is that useful, and then later I realize, oh, that paper is really important, and so then I can find it in my database. Now, it's true that there are databases like PubMed online, and they're very useful, but they don't contain the extract from the full text, which I include. And I also categorize the papers. So for SARS, I have a category for ventilation, for transmission, for antibody testing, for PCR testing, things like that. So if I want to look at it, I can.
Is that database available online?
Well, to some extent, like I have some query interfaces where you can go into the database and do queries on it. So
it's inspiring to me that someone who doesn't have a medical background, although you have a scientific background, can get to your level of scholarship just by going back and reading original studies and all that.
Yeah, it took a while. Like, there was a lot of terminology I didn't need. I had to learn it. But I've worked with a lot of top scientists like Peter Duesberg, David Raznick, Marco Ruggiero, and a fair number of doctors as well. And so I've learned a lot from them as well. There are so many people I owe so much to, but I seem to be in the position where I'm kind of in the middle, where I have enough knowledge that I can write about these things. And I have maybe more time than some busy doctors and scientists.
One thing before I let you go, David, you're an expert witness for the telecom companies. So I wanted to ask you about 5G, because I noticed now there's 5G masts in my neighborhood. And we were talking a little bit about this before, and you said they might not even get it up to 60 gigahertz.
60 gigahertz has been accused of interacting with oxygen, and it does. But the interaction is a very weak interaction. Radio waves are not ionizing. On the EMF spectrum, at the top end, you've got like gamma rays, which are extremely dangerous. They're ionizing radiation. Underneath that, you've got ultraviolet. And we all know that you can't see it, but if you're in the sun for a long time, you can get seriously burned by ultraviolet. And then there's visible light. And then less energetic than visible light is the radio spectrum or microwave spectrum, the same thing. Now, it's true that if you have enough energy in a microwave signal, like in a microwave oven, you can cause damage. You can imagine that if you put your arm in a microwave oven, if you could, that it would cause severe burning of some kind. It would not be a good thing. But a microwave oven operates at 600 to 1200 watts, and the average power output of a cell phone is 0.01 watts. So you've got like between a 10,000 and 100,000 difference. So with EMF, the whole thing is energy, and how much energy do you have? I think that if you were to spend a lot of time in a major radio hub, you could develop some health problems from the amount of energy that you're being exposed to. I agree with that. But as time has gone on with 3G, 4G, 5G, the amount of energy used has gone down. And the reason for that is a little bit surprising. I mean, first of all, it saves battery life, but that's not the main reason. The problem is with so many people communicating, they interfere with each other. And the way to minimize interference is to lower the energy of the signal. And so the 4G, 5G engineers do not want high energy levels. Like if you're one person, you could say, I'm going to broadcast at high energy because then I get a clear signal, but then you ruin it for everybody else. So the systems are always modifying the energy. As you move closer or further to a cell, the energy goes up and down. So the actual energy is relatively constant.
What about these satellites that SpaceX is putting up to beam 5G down on the Earth?
Well, I'm not sure about the specifics of the amount of energy, but usually those things are probably transmitting at quite high power in space. But by the time the signal gets to the Earth, it would be at relatively low. Because if it broadcasts at a high energy, first of all, you wouldn't be able to power the satellite. It would just be too much energy. And secondly, you'd interfere with everybody. So the amount of energy in your phone won't be that much different. Now, it's true that for a satellite phone, your phone will have to broadcast at a higher energy level. So 0.01 watts is not going to do it. So I'm not going to say that those satellite systems couldn't be somewhat of a problem. I think they're probably impractical. There was a spate of satellite systems in the 1990s, and every one went bankrupt. And so I suspect the same thing's going to happen. The problem is that it's really hard to satisfy a lot of customers from a satellite. You have a limited amount of bandwidth, and so it becomes very expensive.
Could the satellites actually be responsible for heating up the Earth if they're beaming microwaves down at the Earth?
I think if you look at the energy they're producing, that's not plausible at all. Okay. I mean, let's say that a 5G tower is broadcasting at 1,000 watts. Okay, so that's equivalent to 10 100-watt light bulbs. So now imagine that you put 10 100-watt light bulbs on top of a 20-meter tower. How much heat are you going to feel down at your level? You're not going to feel anything. So you could actually have quite a high energy level from a tower, but the energy that's going to reach you is still going to be low because it's a sphere, right? So the energy dissipates by the cube. So every time you double the distance, so 1 meter, there's a certain amount of energy. 2 meters, there's an eighth of the energy. 4 meters, there's like a 64th of the energy, right? So it diminishes extremely rapidly just by the laws of physics. So I'm kind of complacent. The other thing is that, you know, this obsession with 5G and radio waves is taking away from the problems that we know are problems. Pharmaceutical drugs, pesticides, poor nutrition, you know, particulate pollution, chemical pollution. There's just so many factors that are causing ill health in the modern world. And the people who are really promoting 5G seem to want to take over the entire world of problems. So they want to explain everything. And so it's kind of like, there's no problems with chemicals, there's no problems with nutrition, there's no problem with pollution or anything like that. Everything goes down to that 5G tower across the street.
Yeah. Well, David, I want to say that it's been such a pleasure and an honor talking with you today, and keep doing what you're doing. And I want to thank you so much.
Thank you, Patricio.
And stay cool.
Okay, yes, you too. Thanks so much. It's been great talking to you.
Bye-bye.